The genetic code for the construction of protein, the basic building block of life, is stored in the DNA molecule. The language of the code is a sequence of nitrogenous base pairs, bridging the center of the helical molecule. This code seems to be partly protected from chemical attack inside the twisting sugar phosphate rails of the molecule. To pass on the genetic code, DNA unzips into single strands. Along sections of the DNA strand, the messenger RNA is transcribed. The genetic code is carried out of the nucleus by messenger RNA to sites of protein manufacture, the ribosomes. There, transfer RNA acts as the intermediary between the nucleic acid language of DNA and the amino acids, which float free in the cytoplasm. The transfer RNA molecule has, at the acceptor end, a triplet of open nucleotide bases, which, depending on the molecule, codes for one of the 20 amino acids used in the synthesis of protein. By linking this codon to a free-floating amino acid in the cytoplasm, transfer RNA acts as a vehicle for transferring the amino acid to the assembly site, the ribosome. The looped end of the transfer RNA molecule, the anticodon, interacts at the ribosome with messenger RNA. The amino acid passenger takes its place in the building of the protein chain. While messenger RNA and transfer RNA both perform vital tasks in the process of protein synthesis, the machinery which actually assembles the amino acid chain of the protein is found in the ribosome. A key building block of the ribosome is the third kind of ribonucleic acid, ribosomal RNA. The largest dimension of the ribosome is about 1/10 the size of a wavelength of visible light. It consists of two subunits, the smaller of which contains one RNA molecule in association with one molecule from each of 21 different proteins, and the larger of which contains two RNA molecules and 34 proteins. This complex little mechanism receives the genetic instructions and reads them. The mechanism also ensures that the proper sequencing of amino acids is followed as the protein is assembled. Protein synthesis begins with the binding of 3 protein molecules called initiation factors to the smaller ribosome subunit. These are followed by the addition of a molecule of guanosine triphosphate. And finally, the messenger RNA strand, which is a long chain of nucleotides. The guanosine triphosphate provides the energy for protein synthesis. Now let's change our perspective slightly. Watch as a transfer RNA molecule approaches with its amino acid passenger in tow and binds to a specific site on the ribosome complex designated the R site. The unbonded nucleotides of the anticodon loop bond to the correct nucleotides on the messenger RNA strand. One of the initiation factor molecules is dislodged from the ribosome, and stresses in the molecular bonds cause the transfer RNA to flip over and bond to the ribosomal complex at a point designated the P site, whereupon the GTP molecule breaks up and is discarded along with the other 2 initiation factor molecules. The next stage in protein synthesis is called elongation. It consists of a cycle that is repeated with each new amino acid that is added to the growing polypeptide chain. 2 proteins, designated elongation factors and 2 molecules of GTP assist in the elongation process. As each new transfer RNA molecule arrives, the nucleotide triplet on the anticodon loop of the transfer RNA links up with the next unread codon on the messenger RNA strand and binds to the ribosomal complex again at the R site. The transfer RNA flips sideways so that the new amino acid is aligned with the old one. This flip of transfer RNA plays a part in ensuring the messenger RNA code is correctly read. The action of flipping appears in part to serve as a test of the messenger RNA anticodon bond. If the codons involved are not exactly complementary, bonding is insufficient to withstand the stress of the flip. If the codons are complementary, the flip is successful. Now the growing polypeptide chain is passed to the new transfer RNA molecule, where it binds, losing a water molecule to form a peptide link. The naked transfer RNA on the P site is released, and the new transfer RNA with the attached polypeptide chain takes its place. When completely synthesized, most proteins are more complex than a single strand of amino acids. They become interconnected with bridges and side chains to produce complicated tangled molecules, each with an engineering design for a specific function in the organism. The ribosomes seem to be ideally located in the cell along the endoplasmic reticulum. This complex canal system allows proteins to be easily carried to other locations inside the cell and out. The recreation of life from a DNA blueprint is an exacting process. At every step, the complementary codes must match exactly, from DNA to messenger RNA to the ribosome, where the ribosomal RNA must engineer a correct link-up. A correct link-up between transfer RNA and messenger RNA. This ensures the correct alignment with the amino acid, where it can be added to the growing protein chain. An accidental change in a single nucleotide base pair in the original DNA code creates a mutation. By far, the greater number of mutations are harmful or fatal. But as dangerous as alterations in the DNA code may be, it is through such minute changes that an organism is believed to evolve. From faulty blueprints, new proteins are born to take their place among the countless variety of the essential building blocks of life.